A year in Principles of Biomedical Science. In the Venom Lab, we tested how temperature impacts the time it takes for antivenom to work to denature venom. Based on the evidence, we concluded that hot water works faster because it speeds up the chemical reaction by causing the reacting particles to move more quickly and collide more often. In this class, Mr. Sharp set up a crime scene for us to perform multiple experiments to conclude how Anna Garcia died. Our testing included vomit testing, blood testing, a toxicology report, and much more. Through these investigations, we learned about topics like macromolecules, protein synthesis, DNA replication, and diabetes. When investigating the crime scene, we learned about different crime scene search methods like the line, grid, zone, spiral, and wheel or ray method. This way, we could pick the proper search method based on the environment and the room layout in order to best examine the scene. In the blood spatter experiment, we tested how the height of the blood dropper affects the distance of the blood droplets from the center stain. Overall, we found out that the radius of the blood spatter increased as the height above the ground increased. By doing this, we could better investigate the blood at the crime scene. To test Anna's stomach contents, we used iodine to test for polysaccharides, Benedict solution to test for monosaccharides, and burette for polypeptides. The vomit turned different colors if the macromolecule was detected. We concluded that Anna's stomach content contained polysaccharides and polypeptides because of the change in color showed positive results.
Another lab our class had to perform was a sheep heart dissection. The purpose of the dissection was to determine the different parts of the heart and show the different flows of oxygenated and non-oxygenated blood through the heart. To represent the blood, my class used red string for oxygenated blood and blue string for non-oxygenated blood. We then took the two different strings and threaded them through the parts of the heart where the two types of blood would normally travel. In another part of the lab, we also had to label all the different parts of the heart. The purpose of the board game project was to demonstrate our understanding of different heart surgeries a person may have. Some spots on the board game represented helpful heart surgeries that could help move you forward, while other spots had disadvantages like clogged arteries that could move you back spaces in the game. In one laboratory experiment, we had to learn phlebotomy. Phlebotomy is a procedure of removing blood from an arm, normally for laboratory testing, using a needle. To complete this lab successfully, we had to locate the correct vein on a fake arm and take the correct amount of blood. We then had to put that fake blood into a centrifuge to represent the separation of parts of blood in a real situation, where it should separate into red blood cells, platelets, and plasma. In this class, we also did a patient zero experiment. A patient zero is the first person to carry and transmit a certain disease. In the experiment, everyone started off with a clear liquid in a cup, but one contained a clear chemical that would turn pink upon contact with another certain chemical. Once everyone had their clear liquid, each person had to switch fluid with one other person and record who it was. In the end, each person in our class switched fluid with 9 other people and recorded their names. Our teacher then placed a small amount of the fluid that turned the certain clear chemical pink into every cup. We then recorded every cup whose liquid turned pink and attempted to find the original chemical holder, or patient zero. Using the Gram stain on bacteria was meant to identify bacteria and the configuration of bacteria by determining whether it was gram-positive or gram-negative. Depending on the bacteria, a cell wall would either retain a violet color when exposed to crystal violet, iodine, and alcohol, which is considered gram-positive, or lose the violet color and retain a pink color from the saffron encounter stain, which is gram-negative. The results at the end would help show the composition of bacteria and aid in determining its type. Everyone in my class performed the Gram stain on bacteria and looked at the results under a microscope.